The first thing you have to understand about homologies is the word homology. And homology are similarities between structures or sequences that are derived, they come from, right? Genetic relationships. Homologies tell you something about the relatedness of the different individuals or populations. So when we look at similarities between structures and sequences obviously by now everyone understands that we're talking about DNA sequences or RNA sequences, but homologies are similarities between two different individuals or populations or species or genus or taxa of any kind that tells you something about how related they are evolutionarily, genetically, and therefore evolutionarily, right? So here we're talking about evolutionary relationships. Now, there is something else I think is worth discussing, that another group of similarities called analogies. Analogous structures are structures are structures that are similar, just like homologies, similar structures that don't have an evolutionary relationship. There is no genetic link. No genetic link. So what would be an example of an analogy? Homologies we're going to talk about in a minute, in the next couple activities actually, but when we look at analogies, what are some examples of that? Well, we can look at a bird and a bat. Bird and bats both have wings, and there is some evolutionary relationship there as far as the two. But you can then look at versus a butterfly. Birds and bats, the wings are very similar. We'll look at some examples soon. But for a minute, let's look at these structures here. Let's look at humans, cats, whales, bats, and birds, actually. And let's look at their relationship to one another. Look at the structures that are similar in all three. You have phalanges, metacarpals, carpals, ulna, radius, and humerus in a human bone. Cats have very similar, you see they color-coded the bone structures. The difference is that cats have evolved to have longer phalanges in certain parts, and other parts of the phalanges are shorter. The metacarpals are longer and thicker, so that the cats can use, they have paws instead of hands. These differences in the bone structure allow them to use that cat paw differently than we would use. It allows us to use our hands as tool makers. Their carpals are very similar, maybe a little thinner. Their ulna and radius are very similar to ours. Their radius extends out a little further, and their humerus is a little shorter. But cats are stockier, and they've adapted. They've evolved to use their their structures, these structures, to walk on, while we evolved to use them to grasp. Whales no longer use these, the ulna and the radius, for walking, so you'll notice that theirs are shorter. But they're using their phalanges, which are much longer, as the structure upon which they can base their fins, right? The way they propel themselves through the water is whales use their hands. You see, these are their arms. This is the humerus. This is the upper part of their arm. It's very short. That their fingers are really the extension of their whale, So these flippers, right, are the whale and dolphins use these, and dolphins have similar structures, but to propel themselves through the water. So their structures are different. They've evolved, but you can tell that they've come from a similar source. You can count the numbers are the same, their positions are the same. The difference is, and we have fossil evidences, by the way, we have fossil evidence that links the whale to a land creature that's actually very similar to hippopotamus. And in fact, the hippopotamus and a whale share a lot of their evolutionary, or have a strong evolutionary relationship, and we'll look at that later. But when you're looking at cats, humans, whales, and bats, they all have very similar structures, and they're just have evolved to use them differently. A whale uses them to swim, a bat to fly, a cat to walk, and a human to grasp. So even though their structures are homologous, they're being used differently. Now, often analogous structures are used in similar way, the way wings are used in bats and birds, and wings are used with butterflies to fly. Butterflies' structures are very, very different. Their wings, butterfly wings, are made of little scales, little, it's a membrane. If you've ever seen a butterfly unwrap, if you've ever seen a butterfly wing unwrap, what you find is that when it was a caterpillar, it cocoons, and over time it changes, it metamorphosizes. That's the word we use, metamorphosis. It changes from a caterpillar to a butterfly. It changes the number of legs it has. It changes almost everything about it. I mean, I don't think you can look at a caterpillar and say, wow, that's what I can tell that was going to become a butterfly. But the point is that the wings were very leathery. They were bendable. They fit inside the cocoon. Then the butterfly unwraps them and then sits for a while, some amount of time, for the wings to dry as they take on their permanent shape. They've unwrapped. They have their shape, and after they're dried, the butterfly can start flying around. And that's the truth of most insects that fly. They're going to do the same thing. They're going to have their wings are made of these things. They look like little scales. And then when they dry, they become stiff and they can be used to fly. So that's why when you damage a butterfly or a fly's wings, it's they can't fly anymore. They can't repair. It's not like a bird's feather. You pluck a bird's feather and it'll regrow. Butterflies are going to be permanently damaged if you damage their wings. There's very little ability to replace a lot of their structures. So butterflies have a completely different way of flying. They have no genetic relationship, they develop flying in a completely different genetic lineage. Yes, we share, insects and humans share a common ancestor, but long before either mammals like bats or dinosaurs like birds developed the ability to fly, or insects developed the ability to fly, they had a common ancestor. Then somewhere along the line, these guys developed the ability to fly, and they developed a way to fly that's completely different. Even though all three of these are flying, birds and bats share structures, homologous structures. Here's an example. Here's the bird structure. Here's the bat structure. Notice the phalanges, the metacarpals, the ulna, the radius, the humerus. Birds have these bones. Here's a chicken wing. I'm sure a lot of you have eaten them. Well, they have the same bone structure as a human, and a horse, and a dolphin, and a turtle. So this is a common structure of quadrupeds, of vertebrates. We share, of quadrupeds specifically, right? These are all the things that evolved from a common ancestor that had these bones. So we have this evolutionary relationship, this common way of doing things. We all use them for different. Dolphins swim, turtles crawl, horses run, humans grass, birds fly, bats fly, and we all do it differently, but we're using the same tools. We just had some got longer, some got shorter, and depending on whether their ancestors went shorter or longer, those were kind of the parts of the characteristics or the traits that allowed them to eventually add them up over time to become the species that they are, the taxa that they are. So horses are horses in part because of how they evolved their bone structure. Same with humans and bats and birds and turtles and dolphins and whales and cats. Insects like the butterfly took another way. They have an analogous ability to fly. They have analogous things called wings. Birds, bats, and butterflies all have wings, but butterflies are a completely different evolutionary branch, where birds and bats share a common lineage, a closer relationship. All right. So when we're talking about analogies, they are structures that can be used for the same function, but they don't have an evolutionary relationship. So butterflies have wings, bats have wings, birds have wings. Birds and bats have homologous structures, their bone structures specifically. And butterflies, however, have wings as well, analogous structure called wings, but they don't have, those wings are not evolutionarily related to birds and bats, the trait wings. So that's the difference between analogy and homology. So as we're moving forward and we're looking at these things, you need to try to keep that in mind, that there are these structures that we'd look at them and you say, well, they're the same. They have bird. They must be related to birds because they can fly. Well, that's not true. And I can tell you I've had more than one discussion with students that wanted to discuss how is it that penguins are birds since they swim, they don't fly. Yes, that's true. They swim. They use their wings like flippers. But they're not dolphins, they're not whales, and they're not fish. right? So that's the difference between analogous and homologous structures. So birds use their wings to swim the way dolphins use their flippers to swim. It turns out that birds and dolphins are genetically related, but so do fish, right? Fish also use their fins or to swim around, very similar to flippers, right? But penguins are not fish. They're not fish that can breathe. They're birds that can swim. So swimming is and flying is not the characteristic that defines birds. Swimming is an analogous ability, not a homologous ability. So let's take a look at homology. So we used to use DNA hybridization, and the way that we used to do it, to compare DNA among humans and other creatures or two creatures together, is we would take the DNA of a human. As you know, they're hydrogen bonded together, right? So that they're easily separated with very low energy. We can separate the strands by increasing the temperature without breaking up the sequence, so the sequence stays the same. You know by now that this sequence is opposite this one, so they're complementary, right? So they complement each other. Everywhere you have an A here, you have a T here. Everywhere you have a T here, you have an A here, G, C, A, T, G, C, etc. That sequence defines the sequence on the other because they have to match, right? They're not exact, they're not the same, they match. A goes to T and G to C. The same with the chimpanzee DNA. So we can increase the temperature of a sample and make them single-stranded, and then we can mix them together. And if this DNA, after we've cut it into shorter sections, because if it's really long, they're not going to match, but we can bust them up into small sections like 600 to 800 base pairs, and we're talking 3 billion base pairs in human DNA, so that's a lot of little pieces of DNA. And then we can let these little pieces of single-stranded DNA between chimpanzees and humans come together and form a hybrid. Half the DNA is chimpanzee, the other half is human. And we can see how closely do they match. So they didn't match here, and they didn't match here. All the rest matched. We call this hybridization. We can test and see how closely does a DNA sequence in a chimpanzee match to a human by doing this form of hybridization. That's what we used to do. Today, that's before we could sequence DNA well. After the Human Genome Project, we developed a lot of technology that allows us to sequence DNA really quickly. In fact, today I'm not sure what the fastest method, and yet staying accurate, method of DNA sequencing is. According to one source, a couple sources, we can sequence the entire human genome, right, in 1 hour today. It took a long time. It took 10 years to sequence the human genome originally. It's a long, painstaking process. Today, it can be done in an hour. And so what we do today is instead of going through this hybridization process, we just sequence the DNA of the chimpanzee and the human or what have you, and we compare those sequences to each other. So either way, you get a measure of how closely related the two organisms are. Their DNA, right? The very stuff that makes them who they are, how closely related they are to another. And we can look at the different primates here. Let's take a look at the DNA similarity between different organisms. Now these are homologies. These DNA, as you know now, at least I hope you know, DNA is inherited. And so if two creatures share DNA, they must have had a common ancestor. I mean, where did you get your DNA? Your parents. Where did they get their DNA? Their parents. And so we're able to group things by how similar their DNA is. Creatures like humans we can do, but we can also do all creatures, anything that has DNA. And almost everything uses DNA. In fact, I don't know of any creature that doesn't. Well, there are some viruses that use RNA, but they're not, theoretically they're not alive. Some people would argue they're not alive. So they're just structures. They're particles. So the word we use is viral particles. They're DNA or RNA in a package. But when we look at creatures like humans, chimpanzees, gibbons, monkeys, and a galago, I'm not even sure what a galago is, but it's a primate, obviously. But here you have these, the capuchin monkey, which is actually very cute, by the way, and the rhesus monkey, also very cute. Don't have them as pets. They take a lot of effort and they live a long time, so be careful. I know some of you were talking about getting a monkey as a pet. That's just not a good idea. When you're talking about a human, is 100%, obviously humans share 100% DNA sequences. And when we say 100%, does that mean that everybody's sequence is exactly the same? No, that means that the genes, we have the same genes, they're in pretty much the same position, that kind of stuff. So chimpanzees share 97.6% of their DNA, the DNA similarity with humans. Gibbons share 94.7%. So you can see that chimpanzees are the most closely related of the primates. And when you compare primates to other organisms, to humans, you see that their DNA is further separated. They don't have as much similarity. We don't have as much similar DNA. Our DNA is not as similar to other organisms as we are to primates. Well, that should make sense, right? That should make sense because when you're looking at creatures like primates, we all have very similar structures. We have very similar structures, and we have these structural homologies, right? We have thumbs, we have phalanges, we have ulnas, just like these organisms do. But our structures are very much more similar to one another than, for instance, they are to a whale or a bat. So our structures are similar, so it makes sense that our DNA would be very similar as well. But that tells us that we are related to we are very closely related to these things we are part of this primate group. We would call this a taxa. And we could put these on a chart. We call this a cladistic chart, right? We call this cladistics, where we start to define evolutionary relationships. We could take a look at birds. We can look at a shoebill, a pelican, a stork, a New World vulture, an ibis, and a flamingo. And we can compare their structures. We could use structures. We don't have to use DNA. In fact, for the longest time, we used fossil evidence and comparative anatomy. And only in the last probably, 30 years have we started to use real DNA evidence to start to compare their relationships. But when we look at the DNA differences, like we can actually score them and the more different they are, the further away we would put them. And as you go down the chart, you get older ancestors. We find that storks and New World vultures share a common ancestor. Further back, their species are older, right? Pelicans and shoebills have a common ancestor. See, every time they connect here, every time you see a connection in this chart, is a common ancestor. So they have a common ancestor that one group of mutations led to a pelican, another group of mutations led to shoebill. They were isolated and they became genetically isolated populations that finally became species. These mutations that collected in the pelican makes a pelican, and those that collected in the shoebill became a shoebill. Same with this common ancestor here. And you can keep going back. There's a common ancestor here that connects. All four of these have one common ancestor that they share with the ibis. You see that? And then the flamingo shares a common ancestor, one of the older species of this group, millions of years ago, go back 50 million years. 50 million years ago, the flamingo had a common ancestor with all these birds. So they went off on their own. They had their own evolutionary a group of genetic mutations that led to the long legs and the long neck and the long curved bill, the classic flamingo look. That flamingo had its own genetic mutations and grouping and genetic isolation that led to this species 50 million years ago. I'm sure there have been changes since 50 million years ago, but pretty much they have their own species that's lasted this long. They've been very successful. The more recent mutations, the pelican and the shoebill, came along a lot later, 10 million years later. So we can actually chart them and chart their relationship, and we can actually call, what we do is we call this group here a clade. So this is one group. We're going to call it a clade. So instead of calling them all birds, we can call them a clade. A clade is a group of taxa. So we can say New World vultures and storks are clade. Why? Because they share a common ancestor. Anytime you have a group that shares a common ancestor, we can call it a clade. We can call anything a clade that we want to when we're looking at a chart like this. This is much more useful than calling them birds or calling them vultures or pelicans. We can look at their structures and their DNA similarities and their evolutionary relationships, and we can say, hey, let's call this a clade. Let's call everything that shares, for instance, this common ancestor a clade. All right, so now these are all a clade, and the pelicans outside that clade. So all these guys share a certain set of characteristics that we're going to call a clade, and it's useful for our discussion for now. And later we can define another clade. Maybe we say, let's talk about the clade that includes the shoebill and the pelican, or let's talk about the clade that includes the stork and the New World vulture. So there we have three different clades in one chart, and we can discuss the differences and similarities, and they can be very useful when you're talking about not just evolutionary relationships, but maybe environmental relationships with niches and why, it could be treatment of disease. It could be a lot of different things that this discussion can then help you if you're trying to find if this, for instance, this New World vulture is endangered and there's some kind of virus that's attacking it. Well, where might you look for some resistance that you might be able to give? Maybe you can genetically modify New World vultures by adding a gene that would make them resistant to a virus. Well, and therefore save the species. Well, where would you look? Would you look in a flamingo? Probably you start to look in a stork because that's the one that's most closely, that's still in its clade, the closest relative it has. But then you might look at a pelican or shoebill, and then you might look at an ibis before you go ahead and look in a flamingo. The answer might be in a flamingo, but it's more likely it's in the stork because it's the most closely related to New World vulture. Same with humans. If there's a disease in humans that doesn't impact chimpanzees or any other primate, and you're looking to see what is it that why is it that humans are affected by this disease and not other organisms? Would you start looking at a bird or a bat? Probably not. Probably you'd want to look at a chimpanzee, since it's the more closely related to us, and see what is it that chimpanzees have that we don't that might help us resist this disease. And so it's understanding these evolutionary relationships help us understand the DNA similarities, which can then help us answer questions or solve problems that we would not be able to answer otherwise. All right, so hopefully you understand DNA hybridization and that it directly measures the similarity between DNA sequences. And you already know that DNA is something that that's inherited, right, from an ancestor. Also, something else that we've discussed. So let's talk about let me just lay it out. DNA, you know it's inherited. You know it's specific. Right, so you know it matches specifically. You know it's the two strands are complementary. So you know it's complementary. You know it's specific, you know it's inherited. And you also know there's mutations, that it mutates. We've talked about mutations, talked about the different kinds of mutations. And the mutations are predictable in time, right? So the longer the species is around, the more time that goes by, the more mutations you'd expect. If enough mutations, if enough mutations accrue, if enough mutations build up, so when you have enough mutations, changes in the DNA that make a significant difference and what is the significant difference? That the two populations don't mate, so that the two populations don't mate, whatever that means. So whether that means they choose not to mate because they look significantly different, or that means they can't mate because they have a different number of chromosomes and they can't successfully make a baby that can make babies, etc. Whatever that looks like, if the two populations no longer can mate, then we call that a new species. So you get a new species when you have enough mutations build up in a population that the differences make it so that individuals are no longer attracted to them sexually, or they can't mate physically because they've changed the way they do things, or they can't mate because their chromosome numbers are different and they can't produce offspring that are fertile. Whatever that means, you have a new species at that point. So when you're talking about DNA hybridization, you're talking about measuring the similarity in the DNA. I think you should be able to answer why that would then talk about how closely related the species are. So study a graph showing the results of DNA hybridization between DNA and other primates, which is most closely related. I think you can answer those questions without too big a conversation. I don't think I have to explain that. 3 looks like it's just reading the chart. They don't even ask you any questions about clades, so let me go ahead and erase those markings. Hopefully you understand how a clade is, and we'll discuss it again in a short bit. State the difference in score. I think you can do that just by reading. Here's the score on this side. Here's millions of years ago here, and here's the different organisms, and you can find the different taxa, and you can follow that backwards. On the basis of DNA hybridization, state how long ago the ibis and Ivor vultures shared a common ancestor. Again, you can figure that out on your own by reading the chart. I'm not gonna tell you the answer there, but remember, common ancestor is where the two lines meet. All right, so that would be a common ancestor between flamingos and the rest of these groups. All right, so I hope that helped you, and I hope that you learned something. Remember to try to keep safe. I know we're in the country's in lockdown, or most of the country's in lockdown. At least the state of Ohio is, and many other states. So just keep yourself safe. Keep isolated from other groups. I know you want to hang out with your friends, but you guys love your Instagram. You love that the YouTubers, you love playing video games. Now's the time to do that when you want to socialize. Get some work done. Make this time useful. Keep yourself spiritually, mentally, and physically active. Make sure you're taking good nutrition in. And again, I tell you to keep separate from a bunch of your friends, just so that you don't spread it. Remember, your parents have a higher percent chance of getting sick, but you can get sick, and I don't want to see anyone get sick. I miss you guys. All right, have a good one, and see you next time. See you whenever they lift this quarantine. All right, have a good one.